Okay, welcome to everybody. Today I have the pleasure to chair Flavia Esposito from the University of Bari. And Flavia is currently a full-time researcher at the University of Bari in the mathematics department. And she got a PhD in 2018 under the supervision of Professor Nicoletto Del Buono. And today she will talk about non-negative matrix factorization models for knowledge extraction for biomedical and other real-world data. Flavia, the screen is yours.
Okay, thank you very much, Pasquale, for the nice introduction, and welcome to everyone. And firstly, I want to thank you, all the organizer of PRIMO group, to arrange these meetings and to join all these young researchers, because I think is a very, very interesting option and very, very good things to share knowledge between us. As Pasquale already told you, I'm Flavia Esposito, and now I am a researcher at University of Bari Aldo Moro, where I work with the MIDAS research group. We work on mathematics in data science, that is MIDAS stands for. And today I present you this work that is a joint work with my research group, but also with Professor Nicola Gillis from the University of Mons in Belgium. Okay, let's start. In the screen, you can see a brief table of the content that we are going to see today. We will start with a brief introduction about the data and how we can treat real data. Later, we will focus on NMF, that stands for non-negative matrix factorization, and I will show you a variant of NMF that is called constraint NMF. Later on, I will show you the work that I developed with Nicola Gillis, that is called orthogonal joint sparse NMF. And to conclude, I will leave you with an open problem about the tuning of the hyperparameter in this context. Now, let's think to data. What we want to do is to extract the knowledge from this big amount of data. The data, we can have this data from the device in our home. So from, for example, Alexa, from our personal computer, but also from our smartphone, from satellite. We can have data from everywhere. Fortunately for us, for mathematicians, this data have a structure. This structure, we can analyze this data in this structure by using linear and multilinear algebra. Let's see some examples. So, for example, if you think to a characteristic, so if you measure a particular characteristic among n observation, among n sample, we can see this data like a vector in Rn. If you are lucky and if you are several observations, but from this observation you can see different characteristics, so different feature, you can have a matrix. So you can have n points in a m-dimensional space. And if you are more lucky, what we can see, if we have characteristic that feature from different point of view, we can have a tensor of order 3 or even higher. But at the beginning, I told you that we are analyzing real data. So we are analyzing data from everywhere, from the device. And today we have a lot of this data. So unfortunately, we cannot do our computation by hand, even if it's very funny, but we need to ask some help to the computer. So we need to move to computational linear algebra or multilinear algebra, of course. The first thing that we can do with this data is perform some dimensionality reduction. This means that we can rearrange our data X using an underlining low dimensional representation. And in particular today, we will focus on linear dimensionality reduction technique. Most of you, I'm sure, that already know SPD and PCA, like the most famous linear dimensionality reduction that use the assumption of orthogonality on each factor. But also there exists independent component analysis that use the statistical independence assumption on the column on one factor, and also non-negative matrix factorization that instead use the hypothesis of non-negative constraint on the factors, on both factors, W and H. So how we can choose between these techniques that are very famous in literature. From the beginning, we say that we have to have a look to real data. So let's have a look to this real data. For example, hyperspectral images from satellite. Hyperspectral images actually are tensors that can be matricized, but what is important is that each pixel, or even better, is each caret is the spectral signature of the point that we are looking. And the spectral signature is a non-negative, has a non-negative value. If we want to perform some text mining, for example, and we want to analyze document and the terms in these documents, we can use the vector space representation to construct the term document matrix, in which on row we have term and on column we have document, and each element actually is a pseudo frequency on the term in a particular document. So like a pseudo frequency, this is non-negative too. And in genomic data that we can see later, we can have this matrix in which on row we have gene, on column, for example, we can have sample, but the entries are gene expression lever that for now transmit, but these are non-negative too. So all these data sets, all these examples are non-negative. So what I want to do is to move this particular constraint from the data also to the factor of the decomposition. So I will focus on non-negative matrix factorization. As I told you, we have the non-negativity also in W, that is the basis matrix, that is called basis matrix, and also in H, that is called coefficient matrix. The possibility of move also this non-negativity from the data to the factor give us the opportunity to make an easy interpretation. For example, by looking W, the non-negativity gives us the opportunity of quantify the importance of a particular row element in each factor that we are going to consider. And for the coefficient, we have the possibility to reconstruct the model with additive operations. From a mathematical or optimization point of view, this problem can be translated into a nonlinear minimization problem, in which we want to optimize, better, we want to minimize a particular divergence function. And the most used divergence function are the Frobenius norm or the Kurbach-Leibler divergence. Actually, it depends from the natural structure of your data at the beginning, but there are several objective functions that can be used. The standard algorithm for solving the NMF problem are iterative algorithm, and what we need to do is to decide at the beginning the W and H. So we have to initialize these matrices, and then we can do the iteration of the optimization of W and H according with some rule. Now I want to just give you the idea of a constrained NMF that actually is the model that we will see later on this presentation. When we analyze real data, it can be useful enforce some particular constraint on W, on H, it depends from your problem. So this constraint can be, for example, enforce sparsity constraint to prevent overfitting, or also enforce orthogonality or soft orthogonality constraint. So analyzing this constraint optimization problem means translate the problem of NMF in a nonlinear penalized optimization problem, in which we have here penalization parameter and penalization function. Now, I want to show you the model that I developed with Nicola Gillis and my research group, that is orthogonal joint sparse NMF, that is a particular model to extract knowledge from biomedical data. So before to enter deeply in the model, I want to give you an overview on gene expression data and why it's important to study this data. I don't want to go deeply in biology, but just to make sure that you understand why we are studying this kind of data. So this is in the picture, the central dogma of molecular biology. And I think that most of you knows that all the information of a human being are in the genes. And these genes are encoded into the DNA. This information starts from the DNA and are elaborated, better are transcribed and translated here in the middle with the mRNA. And at the end of this process, this information are then translated into proteins. You can think to the proteins like the phenotype that we can see. So in the best case, the phenotype can be blue eyes, black hair, so good things. But in the worst case, when we want to extract knowledge to study tumors or cancer, these proteins, this phenotype are the characteristic of the cancers. So the assumption here is that if we are able to quantify what is happening in the middle here, we can quantify and detect how much and which genes are involved in this process. So are involved into the phenotype, into the cancer characteristics. Fortunately, we can do this with the help of biologists and biotechnologists that with some experiment can perform, can arrange and can produce this small chip that with some image processing technique can be translated into a matrix. So into a data structure that mathematician can easily handle. So microarray gene expression better are these matrices or this tensor in which, as I told you before, in row we have genes and in column we can have sample. So for example, we can have patients, but also we can have time steps. So we can have a time series. And if we are lucky, if we have a very big lab to process together all this information, we can construct this tensor that is called gene sample time tensor that sometimes are matricized to have a better visualization. And it's on this particular structure that we construct our orthogonal joint sparse NMF model. What we did is to translate some biological hypotheses into mathematical constraint. So in particular, What we did is to construct a new objective function to extract knowledge from this data. We partitioned this function in several parts. The first part was the KL divergence to measure the goodness of the approximation. Then we had several penalization terms. So we use constraint and MF to add biological assumption in a mathematical way. So firstly, we had this sparsity constraint on W. W, I remind you, is the basis matrix, so all the information are in W. And we do this to actually prevent overfitting. Then what we did is add this term to have a smoothness between each basis in each time step. This means that, for example, I remind you that on row we have genes. So if a gene is important in one step, so at the beginning of the treatment with a particular drug, this should be important during all the treatment. And later, what we did is to add this soft orthogonality constraint to make the basis disjoint. So if a gene is important in one basis, this should not be important in another one. So we have this joint property. As I told you before, these follow the standard structure of the algorithm for non-negative matrix factorization. But the difference here is that we have to make an assumption on the penalization parameter. So we have to choose at the beginning lambda, alpha, and gamma without actually any knowledge before. And what we did is to keep it constant during all the minimization process. And for H and W, we perform the update according to some particular rule. These rules were from this convergence theorem that we prove. And I don't want to give you, of course, all the proof, but I want just to tell you that the proof is based on the definition of auxiliary function. For those who doesn't know the auxiliary function, this is an upper approximation of the function that we want to minimize. And of course, these as an easier formulation to minimize. So this is the objective function. So we want to optimize over H, and these results was from literature. Whereas for the optimization over W, we divide the objective function in different parts in this way. And for each part, we found a new auxiliary function easier to be minimized in order to have the previous convergence theorem. Of course, we perform some experiments and on real and synthetic dataset. We use a tensor of order 3 with 19 times 30 times 5, and each slice of X was constructed by the assumption that W is a normalized monomial matrix for each slice with similar variance, and whereas H was randomly chosen. And on the other way, we choose as a real data set one from the database, a biological database, because fortunately with the gene expression, there is a big database in which you can choose the best data for your experiment. We choose this one with 52 genes, about 27 patients affected by multiple sclerosis with, that are treated with protein interferon beta during 7 time steps. Of course, we try to perform some evaluation of our model by using this measure. Firstly, we use this approximation error that is actually a Kullback-Leibler divergence, a relative Kullback-Leibler divergence. Then we use two sparsity measure for W, the one that is the standard Euler sparsity measure that actually is like a L1 norm, a relative L1 norm. And the second one is like a pseudo L0 norm, because it's a measure that counts as 0 all the elements lesser than a particular value. Then we use also a relative distance between two different slices for the central term. And for the last term of the objective function, we use this relative orthogonality measure. We, of course, compare our model with the standard existing method, but we had to compare it for all the possible combination between each choice of the parameter. So, for example, setting all the parameters equal to 0, we compare our model with the standard multiplicative update for NMF. Setting just gamma different from 0, we compare the model with the NMF on orthogonal subspace to enforce the orthogonality constraint. For lambda different from 0, we did the comparison for sparse NMF. And for alpha different from 0, we compare our model with the multiplicative update unfolding. That actually is the unfolding of the tensor, but with the same W for each slice. The most important part, the most important assumption here is that we did a static choice of the hyperparameter lambda, alpha, and gamma. This means that we decide at the beginning of the process a value that is this one, the same importance in the objective function, and later we keep it constant during all the process, all the minimization process. Here in this picture, you can see the behavior of the relative error in percentage for each iteration, and you can see the standard decreasing behavior for the relative error. Whereas in this table are the numerical results for the measure that I told you before for the real data set. So we can see, having a look, that it works better in it works good in comparison to the standard technique. Here, at the end, we have the numerical result with all the hyperparameters different from 0, and we can see that all the assumptions are respected. And at the end, map, so a graphical representation of the matrix W, that I remind you, in W there are all the information that we want to extract. And what we did is extract the raw element here that are actually genes, and we try, with the help of biologists, to analyze these genes and to see if these are actually more relevant respect to the other genes involved in the process according to the disease and the treatment with interferon beta. And biologists analyzing biological process and pathway analysis confirm this assumption. Now, what I want to do is just to give you an idea of what we are going to to study in this last period with my research group, with the Midas research group. We are focusing on the problem of, on the open problem of the tuning of the hyperparameter. The hyperparameter, in a general way, it can be the parameter of a model that we have to fix at the beginning of the process. So it does not depend from the learning approach. So for example, the hyperparameter can be the number of layers when we are looking to neural network. It can be the learning rate if we are using a stochastic gradient descent, but also it can be the penalization parameter in a penalization, in a minimization process. So in the problem of constraint NMF, it can be our penalization parameter. Here in equation 4, you can see the standard definition of hyperparameter optimization problem when we are talking about a supervised learning problem. So the hyperparameter comes from the minimization of a particular goodness over the test set, subjected to actually an inner minimization problem over the loss function analyzed on the train set. We are start to studying what is the literature panorama in this field, and we can see that there is the formulation like a B-level optimization problem, in which we want to find actually the hyperparameter by a minimization over a response function subjected to the minimization of the actual parameter of a particular loss function. And in this B-level problem, the most difficult computation is about the hypergradient, that actually is the gradient of the response function. In particular, what we are going to study is to study the approach like a dynamical system. So we are considering the minimization of the parameter, the standard parameter, like a gradient descent. And in this way, our problem is this one in equation 7, in which we want to minimize the response function subjected to the gradient descent. So we have to find the hypergradient with by the chain rule, because we have an implicit dependence on lambda, and with a lot of substitution, but I don't want to go in deep in this kind of stuff, but with a lot of substitution at the end of all this process, what we have to do is to solve actually two dynamical systems. The first one is this one in equation 10, in which we have to find actually the parameter of the optimization problem, and this one in equation 11, in which we can find y that give us some help in the construction of the hypergradient. So this is just a brief overview of the literature panorama on, about our idea that we want to apply on the problem of NMF, on sparse NMF. In particular, we consider again a new constraint optimization, a new constraint NMF that is a penalized constraint problem, in which we want to minimize the Kullback-Leibler divergence plus the penalization parameter and the sparsity constraint measured by the L1 norm. What we did is to find another convergence theorem with the new update for W and H. And at this point, we had two options. So we can do the standard static option that is actually the most used, This means that we have to fix at the beginning the parameter, and we have to keep it constant during all the process. That actually is what we did also with the other model, orthogonal joint sparse NMF. And the other option is to perform a dynamic option. So this is our new idea, to reformulate the NMF problem like a B-level NMF problem, in which we want to minimize the response function with a KL divergence to find lambda, subjected to the minimization of the loss function to find W, that is actually the real parameter of the minimization. So what we want to do is to incorporate the minimization of lambda in all the minimization problem with this B-level NMF formulation. Unfortunately, I have no experimental result to show you, because this is just an idea. We are for now developing just the theoretical part, but I'm sure that in the next month another someone in my group can give a talk to show you the progress of this project. So to conclude what I did, what I show you today, I hope that I convince you that NMF is a powerful technique to analyze big amount of data. And I show you also constraint NMF that is useful when we want to enforce some particular properties when we analyze real data. What we did is to add a look to a model for biomedical data, to microarray data to analyze tumor sample. And this model tried to join biological assumption and to translate this assumption in mathematical constraint. For this model, we developed some new multiplicative update, and we proved that it works very well in comparison to the standard literature of NMF algorithm. And at the end, what I did is to show you the open problem of the hyperparameter tuning and our starting idea to try to solve this very, very difficult problem. I hope that you enjoyed the talk, and thank you for the attention. And if you have any question, please, I'm here.
Thank you, Flavia, for your nice talk. A virtual clap for you, unfortunately.
Thank you.
Yeah, okay. There is also a virtual clap from Monica, and I think from all the attendees. If you have any question, you can open your microphone, or you can write down in the chat, and then I can read the question to Flavia. Well, maybe I can start asking you, what about, well, how do you initialize your H0 and W0 in your process? Because
yes, this is a very, very good question. I show you. Actually, in literature, the most used, I mean, maybe the only used approach is to initialize these matrices by random matrices. But there exists a lot of methodologies to initialize these factors. For example, we can initialize them in a deterministic way by using a variant of SVD that is called non-negative double SVD. Also, we can initialize with some hierarchical clustering, some spherical clustering, also with the genetic algorithm. There are a lot of methodology. In fact, we are collecting, we are trying to make a review of all these methodology, and we are trying to compare the results of these approaches, because the difficulty here is to try, is to see if there is some difference in the extraction of knowledge instead of the performance of the algorithm. So we are trying to arrange a work on this that we are going to submit in these days. So your question was perfect.
Thank you very much for your answer. And are there any questions from the attendees?
I do have a question, if I can.
Yeah.
Okay, so Flavia, you said that data can be considered as 3D tensors. So have you tried to compare your NMF model with some other tensor decompositions, or like non-negative tensor factorizations, or do you want to do that? Thank you.
Yeah. Hi, Domitilla. Yes, this is a good, interesting question. For this model, what we did that is actually the most similar to what you are asking, is the comparison with this one, with this multiplicative unfolding. Actually, this is not a real tensor decomposition, but is what, but is the most similar. But no, for the standard tensor NMF multilinear independent component analysis, we didn't do any comparison because the difficulty here was the choice of the parameter. So this comparison is the most similar to a tensor decomposition.
Are there any other questions? Well, I have another one, if I can. I'm really interested in the application. The only information that you have, the only prior information that you have on your DNA application is that the W vector must be sparse. Is it right? Well, you are using the L1 norm.
Yeah.
So I was thinking that you were assuming something like sparsity on W. Is it right?
Yes, but actually another information, but I don't know if it can be called like a prior information, is are this one that we want that the W are similar in each time step. So we want that the treatment with a drug should be constant during time. So if I give you, I don't know, aspirin, the first time should be the effect, it should be the same than the second, the third, the fourth, the fifth, the fifth, and so on. That cannot be just one time in which the aspirin works. So this is the prior information on these terms. Whereas information here is about the disjoint of the basis. So if we have that a gene, you should think to the basis like a biological process. So the first basis can be, I don't know, the anti-inflammatory process. The second one is another biological process. So if a gene is important in one process, it cannot be important in another biological process. Prior information actually here.
Okay, thank you, Flavia. Are there any other questions from the attendees?
Yeah, if I can, maybe I have a very stupid one, which probably shows how less I know about this kind of problems. So you use the Kullback-Leibler divergence. Maybe I lost it at the beginning. Is there a modeling reason for that? Like, what would happen if you use like the 2 norm? Like, because again, as I said, I'm not very, yeah, if you use the Frobenius norm, does the result change a lot?
We didn't try with the problem in the context of microarray because, as I told you before, actually we choose a particular divergence according to assumption that we have on our data. And when we are looking to gene expression data follow a Poisson distribution and are like counting, so like counting the web click. For this reason, when like gene expression data, the divergence associated to this kind of data follow this kind of distribution. So the Kullback-Leibler divergence is constructed. But of course, for more technical results, it could be interesting to compare the results from the Frobenius norm or other divergence.
Perfect, thank you.
You're welcome.
Okay, I think that we have time for, if there are no other questions, we can thank Flavia for her talk. And I want also to say that the next PRIMO meeting will be held virtually next week. And the speaker will be Luca Ratti from Genoa. So let's see in 1 week. You will receive an email as always. Okay, thank you, Flavia, again, and thank you to all the attendees. Bye-bye.
Okay, thank you. Bye.